Welcome to the Deer Wizard Podcast, conversation shaping the deer industry. I'm your host, Josh Newton, the Deer Wizard. Through interviews, advocacy, and industry news, we deliver field-proven insights to help producers build better herds. Ladies and gentlemen, welcome back to another episode of the Deer Wizard Podcast. I'm excited to have a guest on today that we've spoken with in the past. Many of you know her. She specializes in EHD vaccines. I'm talking about Ashley Peterson, the ISPrime director at Medgene Labs in Brookings, South Dakota. We have an extensive conversation with Ashley about how their technology came to be, what sets it apart from other platforms, and just a really good educational discussion about EHD. This is part of our education platform that we're providing relating to epizootic hemorrhagic disease. We do get into a bit of blue tongue, and it's a really interesting conversation. Again, I am not an expert on EHD and blue tongue, so when we have these conversations, I'm trying to pry and tease out some of that info, and hopefully, if you're watching or listening, that you can benefit from that as well. So Ashley has a wealth of knowledge in the EHD and blue tongue space, in immunology and vaccines, and you'll see, we saw Dr. Wisely here last week on the show. We're going to be getting regular updates from Ashley as well, and so we're going to start building out a channel list on the Deer Wizard podcast for separate topics relating to animal health, one of those being EHD, obviously chronic wasting disease, and lots of other fun stuff in the future. So I really hope you enjoy this conversation, and I want to welcome Ashley Peterson to the show. Ashley, welcome to the show.
Hey, how are you?
I'm doing well. I appreciate you coming on today. I was chatting with Dr. Samantha Wisely from the University of Florida at the Cherry Lab recently. We had her on the pod, had a great interview, and I want to cover some of the other side of the research that she does that has an impact on the deer industry. Before we get into that, can you just introduce the audience to yourself, give a little background, and we'll kind of go from there.
Yeah, I am Ashley Peterson. I am the ISPrime director at Medgene Labs. I have been with the company since 2012, 2014, part-time 2012, full-time 2014. So the company basically started with technology founded by Dr. Alan Young, a professor from South Dakota State University. And he was working on an assay to try to detect scrapie, a prion disease in sheep, using a bench assay versus brain samples. And so when I was getting my bachelor's degree in animal science, I was working at his lab at South Dakota State University. So that's how I got connected to Alan. And then I ended up getting a master's degree while working part-time at Medgene, working on that assay for the scrapie detection. And then went full-time for Medgene after I got my master's in veterinary microbiology. So been doing science-related things with prion diseases for the beginning of my career, I guess, and then to support Medgene with grants, we were working with Kansas State on a vaccine project with Rift Valley fever. That vaccine did really well. Rift Valley fever is a foreign animal disease, and so spinning off from that, and the prion side of things, Alan knew Sean Schaefer, the executive director of NADIFA, and Sean told Alan about EHD. Um, EHD is an orbivirus. The targets for vaccine development are well known. So Alan was like, hey, we could use that as a vaccine model since Rift Valley is a foreign animal disease and we can only do so much for that sort of research. So to continue our vaccine side of Medgene, we started working on EHD. And that's where our EHD kind of launched, and that was in 2014. So we've been doing stuff ever since on that. Um,
Yeah,
so that's how we started.
it's so interesting to see like the backgrounds of how things come to be, right? It's kind of, you get a little bit of right place, right time, and also those, personal and professional relationships that you've made, across your career. And like, so just, like Alan and Sean knowing each other.
Mhm.
Um, EHD wasn't even on Alan's radar, right? And
Right,
so like
exactly.
it wasn't his area, but like
Yeah. And
Sean's like,
neither were
hey,
vaccines.
right. Is it
Neither
it's
were vaccines, but
yeah, that's
that
wild.
we found, so baculovirus expression system is what Medgene uses now, and that works really well. And once we got on the vaccine side of things and realized, we could really make them work well, and Alan started to get a vision of how we could do vaccines differently, especially when there was the outbreak of PEDV in 2014, and he saw that there was a gap in how the USDA licenses vaccines, and it takes a long time to get veterinary biologics from concept to market. 10 years typically, a
Mm.
long time. So tons of pigs were dying. You could technically use a baculovirus expression system platform to get vaccine made in a very short amount of time, but the USDA had no way for a company to get that to market fast. So we actually helped the USDA write new regulations for prescription platform vaccines. And Medgene was one of the first to have a prescription platform license. So from Rift Valley fever vaccine to EHD vaccine, we completely went total vaccines, and now we're swine, cattle, turkeys, doing some fish studies. I mean, we're into kind of everything now. And
You failed to mention rabbits.
oh yes, yeah, rabbits, Josh.
Well, I just
Yes, rabbit vaccines too. Yeah, I mean, we're saving. We're going to have a paper come out about the pygmy rabbits in Washington. Um, they have used the RHDV2 vaccine in those wild populations, and they're now thriving. So, yeah, our RHDV2 vaccine for rabbits is keeping those bunnies alive, so that's good too. And we were actually sought out by the USDA to make that vaccine versus us even knowing that we should go after something like that. But RHDV2 is a virus that kind of came out of nowhere in the rabbit population. It was overseas and then got to the US, and the only vaccines that were available were in Europe. And so the USDA was allowing importation if there was an outbreak. So it was reactive versus preventative medicine. And they did not like having to do that. Also, those vaccines, you had to use liver from infected rabbits
mm,
to make the vaccine. Again, our baculovirus expression system does not use live animals. No animal products are in our vaccine. So they loved our technology, asked us if we could do that. The target for RHDV2 is well known, so we got it into our baculovirus expression system, got a vaccine put together, did the trials, works amazingly well, very effective, and now we're hopefully full licensure anytime now.
mm,
So,
that's exciting. Uh,
yeah.
and just for the folks listening, why I brought up rabbits is that I live in a small bubble in the deer world. Like deer is pretty much all I know. And I had seen Ashley at a trade show that I was attending, and they had a rabbit, a big rabbit. I don't know what kind of rabbits are those. What are they called?
I think that was a Himalayan.
Himalayan. This thing's
Yeah.
huge, right? It's like 20 pounds. And he's like hanging out. And I'm like, why is there a rabbit in your booth? And she told me an interesting fact that I'll never forget. And it's that uh, dogs are the number one companion animal, cats are the number two companion animal, and rabbits are the number three companion animal, with like 6 million rabbits as pets in the United States. And so, if you know anything about uh, being a dog or cat owner or companion animal owner, the folks that have animals as part of their lives are willing to spend any amount of money to keep them happy, safe, and healthy. And so I just never thought of a viable business model relating to, a vaccine for rabbits. Like I just didn't know that was a thing. And of course, that's my short-sighted, naive look at the world. But like obviously, it makes sense. And so Ashley told me that. That's why I joke about rabbits. So anyway,
Josh has
it's
never forgotten
no, and
it.
I won't just because like I just it never like it never crossed my mind that like that was a thing, and it's actually a huge thing, like a massive thing, and like the bunnies all over the world rejoice, and if
Mhm.
you're a bunny owner, I'm sorry. So anyway, super cool on the technology. So like let's talk about like the thing that at least what I think I'm hearing, the thing that makes Medgene unique as a lab.
Yeah, so as a vaccine manufacturer, we obviously operate a lot different than other manufacturing companies. Our goal is to stay very connected to our customers because our platform is meant to stay up to date with the current strains that are circulating, especially on the swine side, like what is currently at the barn, because different strains of rotavirus will come in and they are different, and vaccines don't always cross protect that well. So we have to know what is circulating, get those vaccines updated and back to those barns for proper protection. So I'm not supposed to use that word. But anyways, um, so we have a system which is ISPrime. As I mentioned, I'm the ISPrime director. So what that means is from the herd to the end goal of having a successful vaccine, we monitor that whole process. So we get samples from a herd to identify what pathogens are affecting that herd. So we'd have to do diagnostics at a diagnostic lab. We get a PCR positive for whatever pathogen is affecting them. And then from that PCR positive result, we do sequencing. So we're also different where we don't have to have an actual virus or bacteria. We just need a sequence to a specific part of that virus or bacteria. From that, we compare that specific part of a virus. So for EHD, it would be either the VP2 or VP5 protein on the virus. And we compare those proteins to what's in our vaccine to make sure that our vaccine still has the appropriate, what's called epitopes. So proteins are not linear structures. They're basically big balls, big globs of molecule, like a molecule.
Mm-hmm.
And so the outside of that protein is what is available for antibody antigen interaction. So the antibody will bind to specific sites. Those are called epitopes. So our technology allows us to look at those specific epitopes instead of the entire sequence. So that's what we're looking at when we get the VP2 or VP5 sequence are those epitopes to make sure those epitopes are still the same between the sequence in the field and what's in our vaccine. And as long as those are matching, then our vaccine is still up to date and good to use in the field. If we have some missing epitopes, then that's when we need to either put new, what we call constructs. So that would be the other, so like new VP2 or VP5 in the vaccine to cover those missing epitopes that the current constructs don't cover. That is not common on the EHD side unless we're talking about EHDV2 versus EHDV6. So on the EHD side, there's three main serotypes that circulate in the US: EHDV1, 2, and 6. Currently, our vaccine covers EHDV2 and EHDV6. So if we would look at the sequences of the two proteins from EHDV2, so VP2 and VP5 from EHDV2, those look different than VP2 and VP5. Sorry, VP2 and VP5 from EHDV1. So that's why we have to have those specific proteins from the different serotypes.
All right.
And then it also allows us, if a new strain comes in, we can identify that and get vaccine made and put in to get out to the field.
All right.
When the USDA allows for that.
Sure.
So,
USDA is the gate holder.
correct.
Let's
USDA Center for Veterinary Biologics.
Gotcha.
That's who regulates us. So
Okay.
they're a division of the USDA.
Let's rewind to the 2014 timeframe, and let's start from the beginning of the kind of the development process. I heard some bits and pieces of it, but specifically on the EHD side as it relates to your work today.
As far as
Well, so like 2014 was like when you kind of really started working on the EHD side of things. So like what were the first things that you had to do? You
Sure.
mentioned doing diagnostics and then
Yeah.
some lab work. Like what does that look like for EHD specifically?
Yeah. Okay. So back in 2014, when Sean kind of let Alan know, hey, EHD is a real problem in the deer industry, and we don't have a really good tool for vaccines, at least, to combat this. Alan started to research it. He's like, okay, there's good information, good tools for vaccine targets. So first we went just after EHDV2. It's the most prevalent strain, causes the most problems. For vaccine development, it's easiest to start with just a single target and make sure that that monovalent vaccine works well before you start building on top of that. So our initial research started with EHDV2. We started in sheep because we're in South Dakota. Sheep are around, easy to work with. They're a good ruminant species. Deer are ruminants. So that work started with ensuring VP2 and VP5 were the right proteins to target, which we confirmed. And then we had to obviously understand how much of those proteins we need in the vaccine. There's different adjuvants. So an adjuvant is a component that you put into a vaccine that helps make the immune system recognize that they need to respond to something. If you just inject a protein into an animal, that doesn't really do anything. So for protein or subunit vaccines, you need an adjuvant to get a response. So we had to test different adjuvants to determine which of those worked best for EHD specifically and a ruminant species. And then from there, we just kept building on top of that. I believe it was in 2016, 2017, when we were able to do our first study in whitetails at a farm here in South Dakota. And that also just confirmed that what we saw in sheep also translated to having an immune response in whitetails, because sometimes different species don't show you the same thing, but we did confirm that whitetails responded the same as what we were seeing with the sheep. And so from there, um, in we just kept building data, basically, giving reports to the USDA Center for Veterinary Biologics, and really just educating the deer industry that, hey, I think we have a good tool for you guys, and we're working on this. We would go to the different conferences, present our research, and there was a lot of interest. People wanted to get their hands on the vaccine. And so, working with um, the USDA and with support from the industry, the USDA in 2019 allowed us to provide the vaccine under an experimental label to owners that wanted to have the vaccine. So that was in 2019. In 2020, we updated the vaccine to include EHDV6.
Mm.
So in the background, we were also working on um, different combinations of EHDV1, 2, and 6. Sometimes if you combine different serotypes into one vaccine, you can get reduced performance. So we were testing that. In one of our trials, we did see reduced performance of EHDV2 when we included EHDV1, and that is why we just released EHDV2 and EHDV6 in 2020. Um, there are probably ways now we could tweak the different levels of protein. We also have a different adjuvant where there's two components. So we could put, the proteins for EHDV1 in a different component, because we think it's probably protein interaction that was happening that allowed for that immune response to be reduced for EHDV2. So we would be prepared to add EHDV1 to the vaccine at this point, especially as we see EHDV1 sometimes be more prevalent. Again, that's a more cyclical serotype. It's really not seen as common as 2 or 6 on a yearly basis. But the USDA has not allowed us to change our 2 and 6 vaccine. So until we get to the next licensing step, we're at 2 and 6
Hmm.
until then.
And
So,
those are they're just like way more common than one.
oh
Just
yeah,
just generally speaking. Not to
yes,
say that you can't find one, you certainly can, but
exactly. Yeah.
um,
EHCV2 is definitely the most prominent every single year from data from Cherry. So
mm,
you said you talked to Dr. Samantha Wisely.
yeah,
She's the director of Cherry, I think is her title.
yeah.
Um, they have some nice research showing, at least for Florida, that EHDV1 is seen about every 3 years. And even on those years, it's still the least prevalent of the three.
Yeah, it's interesting. So she had mentioned 2012 as kind of this like um big date, right? And it was where they saw I think she said like 32 estimated $32 million worth of damages from deer farmers as far as loss went in that year. And like I asked her about it, and I'm pretty sure um, it was Hurricane Lee, or at least that sticks in my brain, and that pushed up through Florida. And like I think we got like 15 or 18 inches of rain in like a 24 to 36 hour period at my place, and like it was washing out towns. Shortly thereafter, we had EHD outbreaks in Pennsylvania. And while it's limited,
Mhm.
we still had them, and it's not like a regular thing that people were noticing. So I just thought that was interesting because like that's really about the same time that the conversation popped up with Sean and Alan, right?
Mhm.
Relatively speaking, and then
Yep.
like we
Yep.
have this. So I guess tying into the Cherry Lab, you know, they're doing obviously independent work, but it's all in the same field. What do you like? like, can you use some of their data and can that point you into certain directions around your development with what they're seeing? How does that interaction work?
Yeah, so I think it was also 2019, the same year that we started providing the vaccine directly to the field. Cherry reached out. I had met Dr. Samantha Wisely at NADIFA, I think it was 2019, because then it was, I think it was mid-year 2019 when we were able to start providing vaccine. It was like right in the nick of time being
Right,
able to vaccinate.
right.
Um, and they decided to purchase some of our vaccine and
Hmm.
provide it to some of the Florida deer farms.
Hmm.
And they helped those deer farmers vaccinate, and they collected samples and um, had serum neutralization assays performed to identify if the vaccine was actually inducing a neutralizing antibody response. So what that means is you're detecting actual neutralizing antibodies. So that those antibodies would, if a virus comes in, actually attach and neutralize it. There are other types of antibodies that don't do that exact same thing. So neutralizing antibodies are very important, and a good sign that the vaccine is, sorry, I have to be careful with my words so I don't
Sure.
try to make a claim that we don't have. Um, that a vaccine is very successful.
Hmm.
So they were able to confirm that our vaccine does in fact produce neutralizing antibodies against, at the time it was EHDV2. And then they also monitored those deer and did necropsies on any of them that died throughout the season and then determined what they died from. So did they die from EHDV2? Were those vaccinated? And so from that study in 2019, they showed that the Medgene vaccinated deer, there were zero that died from EHDV2, and the deer that were not vaccinated that they had in the study, I believe, I can't remember exactly how many died, but they showed that there was a difference between vaccinated and unvaccinated, where they support the Medgene vaccine
Hmm.
versus other vaccines that they had done the exact same type of studies and did not see neutralizing antibodies and did not have any difference between vaccinated and unvaccinated
Yeah, that's
groups.
that's
So,
awesome. She was just laying info after info and tell me about, like the how 59 different species of midges they've identified here in North America and the 150 worldwide. And, there's so much to, we'll call it EHD management that
mhm.
kind of goes into things, right? And, it's not to, I think there's a misconception about just vaccines generally that like they
Yes,
provide, pure immunity or whatever you want to call it. And like
right.
it's just not the case. And I think it's kind of testament to what Medgene does, where you're continually, doing diagnostics and sequencing, looking at these protein levels and, making adjustments in real time with
Mhm.
these batches so you can provide the best product to folks, kind of on a yearly basis as we continue to flow through the EHD timeframe. Um, can you talk a little bit about maybe what you're seeing? Because like you probably have the largest eyes on where EHD is in the country and how it's moving around. What's that been looking like over the past, let's just say, 5 years?
Yeah, so definitely EHD is moving north. Every year it seems to go further north, stay there longer, come earlier. Unfortunately, it has to do with the warming patterns. In the South, EHD can be found year round. So over the years, we have been able to develop a better vaccine schedule as we learn through, use of the vaccine in the field that southern states, it does work better if they're vaccinating and boostering every 6 months
Mm.
because outbreaks can happen right now. A couple years ago, we had a Texas farm that had an outbreak in April, like a massive
Right.
outbreak. And like they saw every strain of EHD and some blue tongue strains in April.
Yeah.
So it can be really bad. But the North, for the most part, does just stay on that, August to November peak, but sometimes it starts in June or July when it used to, just start in August. So it's getting, worse every year. And I feel like every year you're like, gosh, it was a bad EHD year.
Right.
And you just keep saying that every year,
Yeah.
um, unfortunately. So, and at NADIFA, the ARS group out of the USDA was there talking about their Midge research, and that was super interesting. And their models show just like infinite numbers of midges when they try to like calculate um, like population models. Like it gives them an infinite symbol. That's not good.
No.
So, I mean, it's just the worst virus, and the vector that transmits it is everywhere,
Yeah.
and at huge populations. So, yeah, just trying to use a vaccine to combat it is not gonna do it. It's not a magic bullet. Just using fogging to combat it is not gonna do it because there's an infinite amount of midges, and you're not gonna be able to kill them all.
Yeah. Um, so I guess from a practical sense, I would think that are you starting to see adjustments in how you're recommending the use geographically to people? You mentioned like an outbreak in April in Texas, right? And so like there has to be some adjustments in the application of the vaccine to give these animals the kind of best shot. That must change maybe timing of when it's applied or administered, north to south and stuff like that. So it's obviously a veterinarian producer driven program, but you're trying to interact with these herd veterinarians and make the best recommendations based off of the data you're seeing and the known science that you have. Is that kind of accurate? How does that work?
Yeah, so that goes back to my comment of for southern states, we highly recommend the every 6 month booster. We know the antibodies that our vaccine induces last about 6 months. So vaccinating every 6 months in the South will maintain antibodies year round.
Hmm.
So for northern states, making sure you vaccinate within a time frame where those antibodies will be around through November is what your goal should be. I know there's people that have to vaccinate bucks in like February. You're not going to have those antibody titers through November, so you're going to have to figure out a way to booster those bucks again to get those antibody titers out past November.
Yeah.
Um, so everyone finds a way to make it work. Um, I know it can be tricky, but I promise you there's people that figure it out. Um, there's a farm in South Dakota that had started our vaccine. I think it was about 2018 when there was a large outbreak in South Dakota, bad enough where Game and Fish had to buy back deer tags because the wild population like got wiped out so bad that
Wow.
there wasn't enough for like hunters to go hunt.
Sure.
So a lot of South Dakota farmers started to use our vaccine then. Well, once it was available. But so they started using our vaccine. They couldn't make it feasible to vaccinate fawns. So we do know that our vaccine, when administered to pregnant does, those does will pass on antibodies through colostrum to those fawns. But the maternal antibodies are gone by 4 months of age. So
Mm.
then those fawns are naive unless they're vaccinated. So that farm um, didn't vaccinate fawns, and they would still lose fawns to EHD. So after a few years of that, and their adults, their fully vaccinated adults would be fine. Um, so they finally found a way to get their fawns vaccinated, and they're good
Yeah.
now. So through experience of using the vaccine, I think people start to see that it works. And I understand from the personal side of, you own these deer and you've heard horror stories from other people of other products that were used or other stories. Um, and so you don't trust the product until you use it, right? So I totally understand that. Um, but we have good data. We have people that have used it for years and stand behind it. So um, I know a lot of people are happy to discuss the use of it and how it's worked for them.
Yeah.
And again, it's not 100% protective.
Right.
It can't stand on its own. Um, I wish it was the saving grace for all of you,
Sure.
but it's just not.
Yeah, it's so interesting. Um you think about the challenges where I'll call it management, right? Where physical handling of these animals to get this, in this case, this particular shot into them. And it's really hard to coincide with the life cycle of the deer, right?
Mhm.
And so that's the challenge, right? But like you mentioned, it can be done. And there's always trade-offs with management. So, you're applying at a vaccine at an inopportune time of year for a particular growth cycle, whether that be, let's just say, a young fawn prior to weaning, or
Mhm.
a buck that has antlers that could be susceptible to damage, or you can come up with, does that are pregnant, etc. But we have pretty well established data that most likely if EHD hits you, the costs of whatever downside that happened during that particular management protocol are outweighed by live animals, right? And so um, it, it's it, it's important to, like you said, find a way,
Yeah.
because it is devastating. Dr. Wisely called it, kind of tongue in cheek, the Ebola of deer. And it's just because, these animals literally just like hemorrhage out from the inside in a very short period of time, like rapid onset disease.
Mhm.
And there's really not viable treatment protocols. Like there's, folks have come up with, farmers are the most intuitive and ingenious people at coming up with ways to save animals, right?
Mhm.
Like that's a fact. I've seen some just crazy stuff, and like these animals live and they prosper. But at the same time, there's not like some like, hey, if you do this, like they live. The virus is just nasty. So,
Mhm.
having a product like Medgene's EHD vaccine is really cool. Before we wrap up, I just want to touch on blue tongue a little bit because it kind of takes a backseat to the EHD virus, and
Mhm.
um, it's confused a lot with
Yes.
EHD, and I want to explore that just briefly, maybe setting the stage for a future conversation. Can you touch on blue tongue?
Yeah, so blue tongue is also an orbivirus. So EHD orbivirus, blue tongue orbivirus, they're cousin viruses and both hemorrhagic diseases, so they cause the same clinical signs.
Mm.
So without diagnostic testing, they can be confused. If you lose a deer, you may think EHD, it could be blue tongue, and vice versa. Um, so diagnostics are very important because we know our vaccine does not cross-protect a blue tongue. We are working on trying to get blue tongue vaccine more widely available. Unfortunately, there are a ton of blue tongue serotypes that are in the US, and trying to get all of that into one vaccine is not feasible. There's only
Hmm.
so much space, and when you have to put so much of those proteins into a vaccine, you can only put so much, right?
Yeah.
So we're working on that. Again, we we have a licensing pathway that we're we're going to eventually have a prescription platform for white-tailed deer, which will allow us to provide more vaccines for whitetails besides just the EHD V2 and 6. And then we could do an EHDV 126 and some blue tongue strains in there. So we can have different combinations, come up with some different stuff, but that's on the licensing pathway that we're on. In hopefully in the next 2 years that will be completed.
Sure.
Um, but yeah, very hard to distinguish those two unless you have a diagnostic test confirming, is it blue tongue, is it EHD? And then you have the cases where you might get a negative for either, and then what? Because it looks like a hemorrhagic disease. There's more orbiviruses out there. Um, there was a Minnesota farm that tested positive for a different orbivirus, a really rare one, um, last fall. So that's without sequencing, they would not have known because it was negative for EHD, it was negative for blue tongue, but it was clearly a hemorrhagic disease that killed this deer.
Mm.
So thankfully, the lab went further and did the sequencing and was able to detect this other orbivirus. So again, with Medgene's technology, we could start to explore those extra, more rare orbiviruses. Cherry has found a ton of unique orbiviruses to Florida. Um, so yeah, there's a lot out there without diagnostic testing. Just assuming it's EHD is not the thing to do. Along Medgene's unique technology, we also someday could look at actually targeting the midge in the vaccine. Medgene has vaccines against ticks and hornflies. We're coming out with a Tyleria vaccine. So we have the technology to target actual vectors. So that is something in the future that we hope to be able to do. So we might eventually have a EHD midge, anti-midge vaccine someday.
I love it.
That's my goal. That's my
Yeah.
goal.
No, it's super cool. I'm gonna make sure that your information along with MedGene's is linked up in the show notes. You know, if anybody has questions about, you know, EHD and or blue tongue, the vaccine, et cetera, they can get a hold of Ashley. She's happy to work with you on that and make sure that she can provide the best insights so you and your herd veterinarian can make the best decision for your farm. Ashley, thank you so much for coming on the show today and sharing your wealth of knowledge with us on this particular topic.
Yeah, thanks, Josh, for having me.
Absolutely. And with that, stay tuned for another episode of the Deer Wizard Podcast.